So good afternoon, everyone. I'm Seeprarani Rath from the University of Toronto. And today in this presentation, I'll be talking about how model-based approaches can be applied to facilitate regulatory approval of topical dermatological products. So during the course of the presentation, I'll first provide a brief background about the importance of generic products, then bioequivalence assessment, and model-based dermatopharmacokinetic approaches. Then we'll look at the objectives of the research, learn about the methodology, and then discuss the results and their implications on topical generic product development. So, why are generic products important? We all know that generic products tend to lower the cost of medications, and this improves the affordability and accessibility, which improves patient compliance, and it ultimately lowers the economic burden of healthcare. And for regulatory approval of generic products, it is important to demonstrate the bioequivalence or similarity of this particular drug product with its reference listed drug. So what are the acceptable procedures? So the usual bioequivalence approaches that are used for oral medications cannot be applied for topical products. And this is because the topical products are meant for local action and cannot be quantified accurately in the bloodstream. So the currently approved clinical method for topical products is the FDA's or the Food and Drug Administration's vasoconstrictor assay, but it is only applicable to topical corticosteroids. So the clinical endpoint studies or clinical trials in patients is the only option. As we saw in one of the presentations, clinical trials are very expensive, they're time-consuming and also inaccurate. So that's why there is a dearth of topical dermatological products on the market, which makes it difficult. So the regulatory agencies like the FDA is urging the researchers to develop surrogate methods. Surrogate methods are substitutes for the actual clinical endpoint studies, which will expedite the regulatory approval of these products. So currently, alternative approaches or surrogate approaches are work in progress. One such approach is what we are looking at today. So it is called tape stripping, which involves removal of unimolecular layers of stratum corneum using tape strip, just like a Scotch tape. This is a schematic representation. It is a relatively non-invasive method which allows us to quantify the amount of drug that has gone into the stratum corneum, and it gives us an indication of the topical bioavailability of that particular drug. FDA had published a draft guidance in 1998, but they had to withdraw it in 2002 because two reputed laboratories had different kind of results, and which was actually due to differences in protocols, but then it was withdrawn. Currently, this method is only recommended in Japan and Europe. So the conventional approaches of using dermatopharmacokinetics involves numerous measurements and assumptions relating to the rate and extent of absorption. And it also is associated with a lot of difficulties in sample processing and analysis. And therefore, we are proposing an approach which involves use of pharmacokinetic modeling to estimate the dermatopharmacokinetic parameters more directly, and thereby circumventing these difficulties that we come across while using conventional approaches. Therefore, the primary objectives were to develop and validate a model-based pharmacokinetic approach to assess the rate and extent of drug penetration into the skin, which will help us to accelerate the regulatory approval of topical dermatological products. So this is the study design where we use around 15 to 20 subjects and where the reference listed drug is tested against the intended generic product. And also, just to avoid any false negatives or false positives, we also include some bioinequivalent products. So the creams or the ointments are applied to the skin, and removed after a certain amount of time, and the skin is sequentially stripped. The tape strips are then extracted using a suitable solvent and then analyzed using chromatographic methods. The observed drug concentrations are used to develop a population pharmacokinetic model, and the data is fitted using suitable software, and the bioequivalence assessment is carried out using statistical approaches. So for model development, there are two types of parameters. One is related to the formulation or the dosage form, which gives an indication of the rate and extent of input, and these are the factors that are relevant to product performance, and it is different between the test and the reference, whereas the DPK parameters independent of formulation factors are the same for both test and reference. This is an example of a model where the skin is described in the form of boxes. The boxes labeled as SC are representing the layers of stratum corneum removed by each layer of tape strip. So as you can see, the first tape strip is discarded because it might account for some kind of residual formulation, but then to account for that, we also incorporate a correction factor. You can see that there is the letters F represents the extent of absorption, and the letters K1 and K2 represent the rate of input. And those are the formulation factors, whereas the K-diff parameter refers to the rate of diffusion, and it is the same for the test and reference product. So the first step is development of the pharmacokinetic model, where the simplest model will have a common diffusion rate across all the layers, whereas the most complicated one, like we saw before, will have an unique diffusion rate constant for each layer. And then it is followed by the analysis of the data using MLEM approach using a software known as ADAPT. And the model is evaluated for its appropriateness, quality, and stability. Then the relevant metrics are computed. So here we compute the rate of input and the extent, which is F, and then the bioequivalence assessment is carried out using the statistical approaches described in the FDA's guidance. And here are the results of one of the studies that we carried out, where we compared the reference with the reference and the test with the test, where you can see that the test by reference ratio here, these are all within the limits that were described. But here you can see there are some numbers that are highlighted in red. That's because the study was underpowered, because we only had 10 subjects, and we know that, like, we computed the power of the study, and we can see that if we increase the number of subjects to 14 or 12, we can pass these limits as well. So, to conclude, the proposed DPK methodology can be accurately used to describe the extent of exposure, and the data is described accurately by the model, and the results were congruent with the conventional approaches that we used. And this is a faster method where you don't have the additional difficulties associated with the conventional approaches, and it is beneficial to accelerate the development of topical dermatological products, which will lower the cost of healthcare in this area. So these are the sources. I'd like to thank my academic host, Dr. Izzy Canfer, my industrial host, Dr. Dusham, and my colleague, Dana. Thank you very much for your attention.